phosphate pathway. So what do you understand by this term, pentose phosphate pathway? Pentose means 5 carbon and phosphate is the group which is attached to the 5 carbon compounds. Hello, I am Dr. Kamya Tikku, and today in this video, we are going to talk about pentose phosphate pathway. Now, what is this pentose phosphate pathway? Whenever you eat food in the form of carbohydrate and the glucose which tries to enter each cell of the body, so that glucose comes inside the cell and gets converted into glucose 6-phosphate. Now, it depends upon the levels of ATP whether this glucose 6-phosphate will continue with glycolysis for ATP production. And if the levels of ATP are high, it may divert towards glycogen production. And if glycogen stores are also full, then it may get diverted towards pentose phosphate pathway for nucleotide biosynthesis. The two main aims of pentose phosphate pathway are, first of all, to synthesize precursor for nucleotide biosynthesis, that is ribose 5-phosphate, and to synthesize NADPH. So now, there are two phases of pentose phosphate pathway, oxidative phase and non-oxidative phase. In the oxidative phase, NADP plus is the electron acceptor, which yields NADPH. Rapidly dividing cells, such as those of bone marrow, skin, intestinal mucosa, they use the pentoses to make RNA, DNA, and coenzymes, such as ATP, NADH, FADH2, and coenzyme A. So we have seen that riboses are required in RNA and deoxyriboses are required in DNA. So that's how pentoses work there. In other tissues, the essential product of pentose phosphate pathway is not the pentoses, but the electron donor NADPH, which is needed for reductive biosynthesis or to counter the damaging effects of oxygen radicals. Now, erythrocytes and the cells of the lenses and cornea are directly exposed to oxygen and thus to the damaging free radicals generated by oxygen. There are two phases of pentose phosphate pathway, as I already mentioned, oxidative phase and non-oxidative phase. Now, in the oxidative phase, glucose 6-phosphate, which has been produced by hexokinase enzyme, it gets converted into 6-phosphoglucolactone with the help of the enzyme glucose 6-phosphate dehydrogenase. In this reaction, NADPH molecule is synthesized. Further, this 6-phosphogluconolactone gets converted into 6-phosphogluconate with the help of the enzyme lactonase. And finally, the enzyme 6-phosphogluconate dehydrogenase acts, which produces another NADPH reduced form and converts 6-phosphogluconate to ribulose 5-phosphate. In the oxidative phase, NADPH is generated. Now comes the second phase, in which synthesis of 5-carbon sugars take place. Just like glycolysis, the pentose phosphate pathway appears to have a very ancient or evolutionary origin, and the reactions of this pathway are mostly enzyme catalyzed. Now, the non-oxidative phase starts with ribulose 5-phosphate, and it leads to production of ribose 5-phosphate. Ribose 5-phosphate is the precursor of nucleotide biosynthesis and helps in making DNA and RNA. Now, this 5-carbon ribose 5-phosphate combines with another ribose 5-phosphate, and it synthesizes a 7-carbon compound and a 3-carbon compound. Transketolases and trans-aldolases help in these conversions. This 7-carbon compound, along with the 3-carbon compound, then converts into a 4-carbon fructose 6-phosphate and a 6-carbon erythrose 4-phosphate. Now, in mammals, the pentose phosphate pathway occurs exclusively in the cytoplasm. And in humans, it is found to be most active in the liver, mammary gland, and adrenal cortex. This pathway is one of the 3 main ways the body creates molecules with reducing power, accounting for approximately 60% of NADPH. The first outcome of this pathway is generation of reducing equivalents in the form of NADPH, which is used in reductive biosynthesis reactions within the cells. Why NADPH is so important? Because it helps to combat oxidative stress. Now, what is oxidative stress? The type of free radicals which are formed inside our body during various metabolic reactions, those free radicals can be toxic to the body. So those free radicals like H2O2 are converted to H2O. For conversion of the free radical H2O2 into H2O, an enzyme called glutathione peroxidase is required. If this enzyme is deficient, H2O2 would be converted to hydroxyl free radicals. But now the role of NADPH is NADPH, which is produced by this reaction, it helps in reducing glutathione to glutathione reductase. Via the reduction of glutathione via glutathione reductase enzyme. And which along with glutathione peroxidase converts H2O2 to H2O. Now, if these free radicals are not converted to this non-toxic form H2O, they can attack cells. Next step is production of ribose 5-phosphate, which is used in synthesis of nucleotides and nucleic acids. Third is production of erythrose 4-phosphate, which is used in the synthesis of aromatic amino acids. Whereas these aromatic amino acids are precursors for many biosynthetic reactions. In fact, dietary pentose sugars, which are derived from the digestion of nucleic acids, they can also be metabolized through this pathway, and the carbon skeletons of dietary carbohydrates may be converted into glycolytic or gluconeogenic intermediates. That's all about the video, and hope to see you soon.